So it's always fun to be over here, and thank you, Benny, and everybody for the invitation to talk to the team. It's also my heart is so happy because I get to see a number of my former students here. So thanks for coming out tonight. I appreciate it. I figure I can't go wrong late in the day talking about food, right? Everybody cares about food to some degree. So that's my goal. The only thing Benedetto got wrong is I'm not mentioning silk in anything I talk about today because it really doesn't play much of a role in food for various reasons I won't go into. I also kept this seminar at a fairly high level, so I'm not going to dig into a lot of hardcore data. I'm going to talk about a little bit of data, but I really want to get the concepts across. And if nothing else, give you a sense of where the future is going and why I think it's so important. And hopefully if I get that across, some of you will get invigorated by what's going on and maybe even start to work in this field, right? That's my ultimate goal here. So as you heard, this is really that we use the term cellular agriculture, but it's essentially growing food from cells, just like a biotechnology process, right? But you leave the animal aside. All you need from the animal are some cells from a biopsy. And then in principle, you never need the animal again. And we'll get into the rationale and the motivations behind it. If anybody's interested, we have a whole program at Tufts. You know, I think it took me all of 12 minutes to come over here. So feel free to come visit, see the center, see what we do, talk to the students if you'd like. The term I like is farming without animals. Usually you do it PH, just so you get the sense it's kind of a segue between traditional farming and the future of farming. Okay, so that's background. The other way to say it very generically is think of these as meat and food breweries of the future, right? So it's animal products made, and that can include meat and seafood and certain kinds of proteins or egg substitutes where you either eliminate the animal from the equation or you at least minimize the animal, and that's where you just need the cells. And brings in a lot of other questions. For example, part of our center that we have in this, some of the folks do consumer surveys, right? And if you think maybe some of you here are vegetarians, right? And you don't eat meat for different reasons, ethical, health-wise, whatever. If you're using cells from an animal, but not harming the animal, does that mean you would still now be a vegetarian, or would you still now eat this new kind of meat? There's no right or wrong answer. My point is these are the new kinds of debates we and others in the center deal with all the time to see what the future holds here. So this is the cartoon version of what we're going to talk about for our limited time today. If we go from left to right here, think of this as sort of the process engineering you go through, right? So left-hand side is pick your favorite animal on the planet, doesn't matter what animal it is, and get some cells from that animal. You can get them from either fertilized eggs, or you could get them from muscle or fat biopsies, depends on what your goals are. Isolate those cells, characterize the cells so you know what you have, and then move forward to the right to fill up some kind of bioreactor to grow from a very small set of cells to a huge number of cells if you're trying to make food, right? Food isn't like making pharmaceuticals. We're talking orders and orders of magnitude scaling difference here, and that's probably the most important message to get across today. And so you can pick different ways to do it. We'll talk about all these things a little bit today in the time we have. And then on the right-hand side, biomaterials come into this as scaffolding materials to give texture, mechanics, et cetera, to the foods you're going to make in combination with the cells. And then on the right, you make products that hopefully a consumer would want to use or consume. Most of the companies in this space, and it varies now with the economy, but somewhere between 150 and 200 companies worldwide operating in some version of this space today. And most of them think on the right-hand side, you have to make things that look and feel like foods you're used to eating if they're going to get consumers to try it. So you see meatballs and hamburger patties and things like that. That's kind of the rationale. So again, that's consumer thinking, whether it's right or wrong, time will tell. And then, you know, there's different versions on the right-hand side. These are all on the top and the bottom unstructured. So, you know, just kind of hamburger and hot dog kinds of things, and then very structured in the middle like chicken. So we'll talk about all those issues more in terms of the challenges and where the field is going forward. So the field itself, excuse me, comes out of the world of tissue engineering, which many of you are familiar with, I'm sure, being in the programs here. And that field is the same thing I just mentioned, right? Finding human cells, though, under the right scaffold biomaterial conditions in the suitable bioreactor to create tissues and organs that you could use to replace or heal human disease and so on. And that field continues on. We work very actively in that field, as Benedetto said as well. But the same tools can apply to food as well as many other technologies. So the idea was to transform the tools we use for human tissues into animal cells for food. And then you have to rethink a lot of the things you're used to doing. So people ask, well, Dave, did you ever try it? The answer is yes, I've tried just about every cell ag produced food that exists, at least in the United States. And here's 3 examples that we've tried. Wild Type is a company on the West Coast. They make cell cultivated salmon. Tastes really good. It's a little, the texture could use a little work, but I'd say the look is beautiful and the texture is good. And we did a tasting of their salmon in Boston a couple of months back, just to bring it from the West Coast to the East Coast. Chicken in the middle, that's the most common product that's made today. And on the right-hand side, Mission Barns, they make cell cultivated pork fat cells. And they make meatballs and bacon and things like this, right? So I told you food is a good topic to get you hungry, right? So the meatballs are great. The bacon has work to do, but they are the 3rd US company just 2 weeks ago got approval to sell their product in the US. So now there's 3 Good Meat Upside Foods, which are both chicken-based, and now Mission Barns, which is pork fat-based. So you're starting to see growth. The regulatory path is tortuous. You have to go through not just the FDA, but also USDA. So 2 levels of scrutiny for safety and nutrition, but those 3 companies have passed. And since I know a lot of you folks go back and forth to Singapore, right? You could buy it in the market in Singapore. It's chicken-based. The challenge here is it's really only 3% by weight cells. So don't be fooled. It's kind of a marketing gimmick if you ask me, but hopefully it gets consumers more familiar with the technology, and that's a good way to start to grow interest, and we'll see where that goes. So that's available in markets. They are not yet in the US. There's not enough scaling yet to do it here. All right, so I just want to make sure we're all speaking the same language. So cell ag, the core of what we're talking about in cell ag is the cells. The cells are where you get nutrition, flavor, kind of consumer friendliness, if you will. There's lots of other technologies growing out there. One is just the plant-based production, which you've all probably seen or tried to some degree. Here, there's really no particular cell component involved. You're just taking your favorite plant material, going through some extra processing. You might hydrolyze, you might extract, then you go downstream in some kind of extrusion or other and make structural products that are sold as foods. And this is out there today. We use this technology in our lab as well. You can see some of the example projects we have going out at the bottom. That's not so germane for today. But what's interesting about this is the reason why this technology took off about 5 years ago is the discovery that you could put heme, particularly from soy plants, into these plant-based burgers, and you elevated the taste. Huge, right? So all of a sudden, these kind of behind-the-scenes plant products, which were not very common, other than vegetarians would eat them, all of a sudden you saw them in every single supermarket on the shelf, and every menu at every restaurant. You still see mostly Impossible Burgers or something like this. And it's all because of the heme, and it's really the iron in the heme, right? When you cook it, it gives it that meaty flavor, that umami flavor, and that's what people like. So the good part for this is it showed this sort of, huge exponential growth in consumer interest and willingness to try alternative foods. At the same time, after they started trying it, what do you see now? Those companies are crashing and burning. And they're crashing and burning for many reasons. Some is the cost is still high. Others are the taste just isn't the same as what you're used to eating as a hamburger or whatever. And third, if you start to look at ingredients, they're over processed, right? Too many ingredients, processing aids, high salt, et cetera. So what I take away from this technology, it's a great start. It shows there's a willingness out there to try new things, but you got to do better in taste and flavor and health. And so that's where cells come into the fore, I think, because this, in principle, should solve these problems going forward. And then, of course, there's a whole field of alt proteins, and people define all these different ways. But obviously here, you're using genetic tools to have host cells produce your favorite protein that you're going to use mostly as an ingredient into the kinds of foods we're talking about. This technology is going well. Again, cost remains the biggest hurdle here to getting more widespread utility. And also in our lab, we have active programs in different kinds of alt proteins for the food space. And you know these, here's a good example, Perfect Day. I always use Perfect Day because one of the co-founders was a Tufts master student with me. They make all kinds of, basically dairy proteins without the cow is the idea. And they have ice creams, yogurt, et cetera, but they're also struggling right now because the cost is so high to make these proteins and scale them up, but we'll see where that goes. All right, so I put all that together, right? If you followed what I said, you've got alternative proteins we just talked about. We have plant-derived materials, those are the blue circles, and then cell ag is what I call the binder. It puts it all together because that's where the cells come from. You need the plants and microbes for media ingredients through hydrolysates and other things. You need them as additives, you need them as scaffolds, and obviously all proteins can be important as growth factors and other mediators of how the cells are going to do in this process. But I think the rest we've covered enough so you have a good sense of what goes in upstream and what comes out downstream. All right, so why am I even wasting my career now on making this new kind of food, right? I had a perfectly happy career studying silk, as Benedetto mentioned, and tissue engineering for human health and well-being. And this is why the students who would come to see me starting 10 years ago, and it could be from any one of the reasons around this sphere, right, that they came to see me might be from biology, from engineering, from the med school, it doesn't matter. There's interest in all these. And some come because they really care about animal welfare and they're concerned about how livestock animals are grown for industrialized meat and agriculture and quality and health. The point of fact is about 80% of all the antibiotics used on the planet today are just to keep livestock animals from getting sicker than they are. And yet you see recalls, bird flus, swine flus every month going on. So you can only pump so much antibiotic and hormones into these systems without having downstream consequences. Sustainability, right? I'll show you a slide in a minute. We just can't figure out how to monitor and control and support a population of 10 billion people on the planet with the way we're producing food today. There's not enough fresh water, land, et cetera. So we have to do things a little differently. We have to think more creatively. That's why biotech and cell ag come into the fore here again. Lots of questions about food safety. I mentioned growing interest in food security, particularly with the new administration, right? And that's why you see a lot of the innovation in this field that I'm talking about today coming from Singapore, Israel, now China, Korea, and others, because they want to be self-sufficient without having to import all their food. And even in the US, we import a ton of food. And so you'll see, I think, more and more interest in how do we bring food production back to the US and not just traditional livestock, but also all the other things many of us like to consume. And finally, food equity is a recurring problem. If you just look at the cities around the US, in the cities, it's very hard to get fresh, high quality, nutritious food today. And there's many reasons for that. We could debate that. But the idea is by generating food the way I'm talking about today, you could decentralize, you can make it local, you can make it large scale. You have many, many modular approaches going forward. And that's in principle the way to address some of this. Okay, and then, so that's globally a little bit and all the drivers why students come to the lab to want to do this. And then from our own US Department of Agriculture, and they're the ones who have also given us a very large grant. We have now 11 academic institutions, including MIT, who work together on the projects to solve the problem of cell-cultured food. And the USDA puts out their reports all the time, and their report tells you already you have to do this. They're looking for alternative, sustainable approaches to food production because the population will not allow you to to survive with needing 30% more water, 50% more energy, and so on. So the drivers are there, but our system as we have it today in the US can't accommodate that growth. And the other thing, I was surprised to learn this, but in the USDA reports as well, almost 90% of farmers have to have a second income today. They can't survive on the smaller farms, just doesn't exist. So my point of saying this is as we go through the talk, you'll see opportunities coming where the farmers would have new ways to generate income that they don't have available today. And that message has to get out better. We haven't done a good job yet, but we're starting to work on that. So the point here is there's many drivers for the interest in this field. To me, the fundamental goal here is to figure out how to make healthier, safer, more nutritious food to feed the broad population. And I think this technology, to me, is the only way I know of to get there. And we look at everything today. So I think it's really important going forward. So the rest of the talk, other than a little bit at the end, I just want to spend time on this slide, because this slide just basically highlights where the challenges are going forward, and then therefore where the opportunities are. And these are maybe a call to many of you out there, depending on what you do for your graduate work or your postdoc work, or when you open your own lab, to think about some of these things, because there's plenty of room for innovation, and we're only at the beginning of this field. It's going to grow slowly and gradually over the next 5, 10, 20, et cetera years. So we've got to figure out how to get costs down, how to scale up, how to make dense foods, not just these loose sausage kinds of foods, make sure we do it safely. How do we get taste and quality there so consumers will want it and want to come back to it? And then will consumers accept GMO or not use GMO? And as a research lab, we do both because we care about both. Ultimately, consumers will decide. In the US, GMO is pretty well accepted, I would say. Obviously, not so much in Europe. Debates on in other parts of the world, but all of this is moving forward either way. One of the arguments, we just put out a paper last year, we argue that if you don't use GMO, you're going to be left behind because it gets you to the finish line much quicker. So you have to, again, weigh all this depending on what your goals are. All right, so back to, this is what I want to walk through with some examples and some challenges from left to right. So I'll start with the cells and move my way through to scale up the biomaterial side and so on, just to point a few things out. And it'll just be a couple slides on each because I want to leave time at the end for some questions if we can do that. All right, so this is what the process looks like, right? We go out to our vet school at Tufts out in Grafton, Mass. We take the biopsy from the cow. You can, again, pick your favorite animal. Come back to the lab, isolate the stem cells mostly, and these are either bovine satellite cells from the muscle to grow muscle tissue or preadipocytes to grow fat tissue, depending on what your goals are. And we take those in the lab, and without going through all the details, we grow up those cells and we see if they form muscle fibers or, bundles and myotubes, and that's what you see at the bottom right. Absolutely beautiful muscle bundles that form from bovine cells, from cow cells. So we know we could go out in the field. We know we can get the animal cells. We know the animal's fine when we're done. It's a tiny little pinprick, and the cells now are available. So the challenge is, now that we can do that, how do we get from that to filling this room with cells, right? That's not a trivial task at all. To do that, we think you have to immortalize the cells. And if you're going to immortalize the cells, you have two choices, right? One is spontaneous immortalization. So you do that by continuous cultivation. You can stimulate with chemicals. You can use UV, and sometimes it works and sometimes it doesn't. And if you can't do that, then you use genetic tools, CRISPR and other. So that's what we found with our bovine satellite stem cells from muscle. We could not spontaneously immortalize. So we went in with some genetic tools. All this is published, so you can, if you want to read details. And the red line here, this is our immortalized cells, just keep growing. That's time in days against doublings, and they just grow and grow and grow. You don't have to worry about them losing their vitality. They don't go through a Hayflick limit. They just keep growing. And the black circles there are the primary cells, and you can see, oh, thank you. It's okay, I can run. Thank you. And the black lines here, those are the primary cells. So you grow them for a while, and they start to lose their vitality because of that limited growth cycle, right? So this works beautifully. We've provided these cells to many, many labs. They work really well. Obviously, over time, if food is made from these, you have to make choices. Do you want GMO or not? But that's what you would have here. And we already talked about speed of discovery, translation to companies and to foods. It'll be way faster with this than if you use the primary cells. Because also the primary cells, the cells here, which means after 50 days and scaling this up, you have to go back to the animal, take another biopsy. Every time you do that, you run the risk of variable cell quality, which means you have to redo your process operation, which means you're out of business. So there are many reasons to think through these approaches, but nonetheless, we do this for bovine cells. At the same time, sometimes you get lucky, or I'd like to think we had some role in it, right? So we went out, I even went out on the deep sea, Gloucester, Massachusetts. We did fishing, caught a cell and caught a fish. This was the mackerel cells that we isolated on the boat. You could see sequencing to make sure they're mackerel cells, just cultured them for a while, spontaneously immortalized, and now they grow forever. And again, these are available to everybody as well. We provide these all over the world. And because it was so easy, one of our spinouts was a company called Wanda Fish to scale this up to make fish food from fish cells, the way we're talking today. So this was just to give you a couple of ideas. We have many other cells from many other species, but that's the general strategy. The challenge here is when you talk about these cells, the first comment you get from the public is, you're feeding me cancer, right? Because these cells are immortalized. How do we know it's safe, right? And so again, I can all day talk about the packages of data provided by the 3 approved companies that show safety data and all that, but that doesn't sway consumers necessarily. They don't want to read those regulatory dockets. So in our lab right now, we're working very hard. This is led by Archana, one of our postdocs, to look at all the cells we work on and look at, for example, this is work in progress. She's just getting ready to write this up, but comparing DNA repair genes on the left and comparing cell cycle genes on the right through many, many population doublings, through storage, stability, et cetera, with the idea to publish all this to say, if you're using our bovine cells or somebody else's cells, this is the stability or not of those cells from a genotypic and also from a phenotypic perspective. So our idea is to build a compendium, a library of this data for every food cell type that people are pursuing, and then make those data available on a website so people can do this. So far, so good with these cells. And we compare this also to human cells used in cell therapies. So we use also CHO cell data. We use sci-fi cells that are insect cells used in pharma. We're looking at cells that are going through injectable, like fibroblasts and other things, human therapies, to compare those stabilities as well to what we're seeing here. So stay tuned, a lot of data. We don't have a good answer. All I can say is the data is very important to have transparent, scientifically validated, not just speculation on what. There's no evidence if you eat a tumor, you're going to get a tumor, right? That's never been proven, but you don't want to have to deal with that with the public. You want to be factual with them. All right, so that's a little bit about cells. I want to shift over to media, because if you look at the last 5 years, I think now there's been 7 or 8 published life cycle analyses of growing meat from cells the way we're talking about today. And every one of them argues that the media costs are going to kill the field. It's just too expensive, right? Because you need things like serum, you need growth factors, and they're all very, very expensive. So without all the details, again, the goal is to remove animal components, so get rid of serum, and reduce costs, so reduce or eliminate growth factors. That's kind of the mandate here, if we're going to get costs in line with scaled up food production, et cetera. So here's what we've done over the last 3 years or so, right? So first we went and developed, this is with our cow cells. I just figured I'd stay with that theme. We do this with our other cell types as well. So here's serum-free media developed for bovine cells. What do? We replace serum with just recombinant albumin. So you can get this from a host of different host cells. So you're not using animals in the equation at all. And you can see the green and the red line there, they do pretty good with days here against cell doublings. Not as good as serum, but really perfectly fine. And 2 other things we found with these cells, and this again is all published. One is still the presence of stem cell markers in the initial bovine cells. So we haven't lost the stemness as we're growing up large numbers. And number 2, once we differentiate, you see really nice myotubes and so on. So the cells immortalized and grown on albumin still behave the way you want these cells to behave. So that's great. So we solved the problem, no animal ingredients. However, those of you who know anything about albumin, it's recombinant, so it's going to be expensive. So we haven't solved the cost, we solved the animal side. So the PhD student went on and he said, okay, let's replace albumin with something that comes from very low cost plant extracts. So we found an oil seed, or he found an oil seed plant that we could do a very simple extraction from. And so we replaced the albumin with a plant extract, and you can see these guys grow almost as good as the albumin one. So now we solved the animal sourcing issue, and now we've solved some of the cost issue by reducing the need for expensive albumin. And again, same stuff over here. We haven't lost the ability of the cells to do what we want. And then the last part is, how do we deal with those growth factors? Because those are recombinant growth factors, very expensive as well. So the student also was pretty clever. So he did some genetic work, right, to express more FGF than what is present inherently in the bovine cells. And he also knocked out or mutated one of the pathways that would affect mitogenesis, the growth of the cells. And so essentially the idea was to have the cells make their own growth factors, so you don't have to add them in, which means you eliminate that cost. So no exogenous growth factors to go in. And it works, right? So again, details being left behind, it's all published. You can see the dotted line up there with the star. That's the holy grail, right? That's the stuff I'm talking about that we changed in the cell, and now the cell is able to grow without having to add FGF as one of the standard growth factors. So we're making progress, but don't be completely fooled, right? If you look on the right-hand side, what you see are these doubling times, right? So as we engineered the cells, all of a sudden the doubling times go up compared to the native cell. So why is that a red flag? That means it's going to take longer to build up enough biomass of cells during your production process. And if it takes longer, what does that mean? It's going to cost more. So we solved the cost of the growth factor, but we didn't solve the production side cost. So this is going to take more engineering, but very doable with what we know on these cells. And we're looking at all these factors as we speak. And there's lots of other things here, but for time, I'll just skip that. All right, so where does this leave us? Just in 3 years, just our lab alone, and other labs are working on this too. So my point is, we started out, all the LCAs say cost is too high. You know, in 3 years, we went from serum containing media, almost $200 a liter, down to, in our hands, you know, almost $15 a liter. So remarkable progress in just a very short time. And the holy grail is to get to less than $1 a liter. And the question is, how quickly will we get there? Some companies suggest they're already there. We don't know that because their data isn't transparent and available. Everything we do, we publish so people can make their own decisions. But my point is, progress has been remarkable in a very short time. So I don't worry about media costs long-term. I worry about other things like scale up infrastructure. We'll talk about that. But media costs are going away. And the other way I present it is, for those of you who know the field, this goes back to tissue engineering days. For anybody in tissue engineering, you know, the iconic picture for tissue engineering is the mouse with an ear on it from the Langer lab, right? You should all know that if you go to school here. If not, go back and start school over. All right, but my point is that that is when the field of tissue engineering started, right? Everyone looks at that as that was a seminal moment to show you could do this, right? You could grow foreign cells and in a scaffold in an animal, and you could create an ear, right? Same thing in this field, in the food field, cellular agriculture. The seminal moment is a little more than 10 years ago, right? And this was in 2013. It was this is a beef burger made from cow cells grown in culture, and it was carried out. The cooking and tasting was done in London, and it was all the rage. It was on the videos, but it jump started this whole field. So think about this. We're only 10, 11, 12 years in for an entirely new technology. And I bring it up here because this hamburger that was grown by Mark Post, who's a good friend, cost roughly $1 million per pound 10 years ago, right? Ridiculous, right? But if you look at where we are a little more than 10 years later, every company and even our own lab would project we're already down to $10 to $50 a pound. So in a very short time, we've orders of magnitude reduced costs. My point again is we're already on the way. It's just nobody invested time, research, and energy in this until you had to, and now you have to. So I think the path, as we say, to truly low-cost media is already within reach. We're making great progress. All right, so that's a little bit, oh, I gotta speed up, sorry guys. That's a little bit about cells, a little bit about media. Very briefly, biomaterials play a key role in all this, as you saw, hopefully in my earlier slide. You can get your biomaterials from all kinds of sources, plant-derived, microbial-derived, and then you can use those as media components or the scaffolds themselves. But with biomaterials, unlike for growing human tissues for tissue engineering, you have to worry about thermal stability because you're going to cook these, right? So very different set of design goals. They have to taste good, and they have to be food safe. So a little bit different sort of guidance here. And I bring this up in many, this is what we were talking about in your office, right? So we're just putting a review out because I think people don't appreciate the challenge ahead just for the biomaterials part of this new technology we're talking about. And I call it supply chain problem, because here are just some of the more common polysaccharides and proteins that are used in food for cell ag today, if you read the papers, right? And here's the annual production in metric tons, estimated, of course, but other than cellulose and some chitin chitosan, everything else is pretty low. And if you just look at food production on the planet, right, we're talking almost 500 million, let me repeat that, 500 million metric tons of meat and related products annually. That includes fish, right? Estimated by 2030. We're already close to that, right? And so even if just 10% that gets replaced by what we're talking about, by cell ag, you're still going to need a staggering amount of biomaterials to support the scaffolding, the texture, and so on. That supply chain does not exist today. So if you're looking at new innovation opportunities, here's a great area for you to think about, innovate in, and get on board. So again, happy to talk about that more. And we do a lot of things in the lab. One example is, I always like to point this out. Somebody will say, well, how long do you need to grow the cells on this scaffold to get the right food? And the answer is, it depends on what you want your food to be, right? So this is a dynamic process, time and culture X. The longer you go in culture, the cost goes up. You can't afford high costs. And here is how much cell, how much biomass. So you have to weigh that depending on the product profile you're after and choose when to stop the culture. And this is just proliferation of the cells, and this is differentiation to make the muscle cells, the myotubes, and so on. So you always have mass balance issues. You always have decisions on time points, depending on what your goals are. And part of this is also just to work on all the standard life cycle assessments with biomaterials to know, are you picking the right biomaterial to match what you need, where are the pain points, and so on. So we have an active program to collaborators at Tufts to look at those inputs from scaffold production, outputs from the kind of food you're trying to make, and what goes into the processing and so on, to at least build models that are relevant to food and see if we can then get a better handle on where we need to focus our efforts. And that's been expanded in many ways. Nicole is my collaborator. She works at the nutrition school as faculty. Together we've published a whole series of papers comparing life cycle sort of assessments with the models on different cell types here. We've compared mammalian cells to insect cells, media components, biomaterials, and so on. So we continue to build models and really try and get quantification of where the factors are that you have to think about here. All right, good. So next is where I think the biggest gap is, and that is scaling. All right, and so as I showed you before, we can do really, really well making muscle bundles, fibers in the laboratory, no problem, right? And those are from the bovine satellite cells I showed you before, or other cell types. We are nowhere near making something like a steak, right? It's not even the same planet right now. And keep in mind, the scale, 200 microns, so that's probably maybe 300 if we're having a good day, versus pounds of food, right? So it's not even close, is my point. And so this is the other conundrum that we face, is most of the companies in the space today think they're going to take what they've learned from pharmaceutical production and translate it to making food. And I think that's complete folly. And you know that. You just look at this picture, you know it's folly. Why? Because first of all, you have a gleaming stainless steel tank, and the picture has more plumbing, and those of you in the field know, process control to get pharmaceuticals produced consistently the way we do it today. And it works great for pharma. Why? Because you could charge almost whatever you want. For food, that all has to go away. You don't have the luxury of process control or tight process control. Even stainless steel, again, there are estimates out there how much stainless steel you would need to build the infrastructure to support food production. It's just almost impossible. So my point again, for at least the students in here, or maybe some of the junior faculty, is, get on board, think with the innovation. How can you grow up these cells with or without scaffolds in new ways that would obviate the need for process control. So you don't have to do all the mixing and all the heating and all these other things that cost money, which means you're never going to sell your product, right? So you have to rethink all that. And to do that, you have to get away from stir tank reactors. And you're seeing companies, there's one right here in Cambridge, ARC, which uses airflow or airlift bioreactors to get away from this. I think that's only the tip of the iceberg. We need to really rethink things differently. So what are we doing? We're changing the paradigm. And those of you who are from the engineering side, you'll understand this. The biggest limitation to the way we do it today is mass transfer, right? That's why process control is needed. So in my mind, get rid of mass transfer. So it's not a limitation. So we do that by growing our cells on fibers, 1D, and on films, 2D, and then downstream processing to make 3D structures, right? So therefore, the cells are not limited with oxygen and nutrients and waste removal. They're in direct contact with the media, and you can do things a little differently. And I don't know if this is going to work. I just think it's a different way to think about it and a way around the current limitations. So we kind of laugh a little bit, but it's true. We're basically knitting meat of the future. That's kind of one of our many types of innovation. And the way it works is this, and we've already shown this works. The first part, it's published, so if you want to read some of it. But the idea is the only thing you need is a bioreactor can be continuous, where you feed in edible biomaterial scaffolding and get your cells to grow as a coating on the surface. And you want the cells to stick pretty well. Because once you've done that, the rest is just traditional textile engineering. So yarning and bundling, where you get dense foods, that's where you're going to get your pork chops and your steaks. I don't know of another way to do it with a systematic approach like this. And then 3D structures can come from all the old school textile engineering approaches. So it's kind of bringing back the old technology, but for an entirely new set of goals. And you can build these devices to be modularized and small. You can imagine a building the size of MIT, right, where you have 5 trillion, edible fibers coming through, feeding cells, all automated, computer controlled to get out the end, the size, density, nutrition, whatever you want for that food. And this could be muscle cells, could be fat cells, and so on. So my point today is this part does work. We haven't scaled it, but we know it should work. As long as this works, the rest will work. I'm confident of that. And keep in mind, for growing food, it's not like tissues for medicine. Here, the cells don't need to survive after this step, right? Once they've coated, you just need the cells for their flavor, their nutrition, and so on. The rest of the cells, as long as they're hanging on there, they can go through the final product and you still get all those benefits. So lots of things to think about, at least we think so. And then the last part is you want to make sure what you make tastes good, right? If it doesn't taste good, no one's going to buy it, even if the cost is low. And so we had a real sort of panic for a while. We're growing all these cells in the lab, fat cells from pigs, the muscle cells. We never knew if you grow a cell outside of the animal, would it smell or taste anything like what you buy for food today, right? If it does, great, but if it doesn't, what do we do, right? So that was a real question mark. So this summary is just, first of all, so if we grow, for example, our pig fat cells and compare it to butcher fat from a pig that we could buy at the local store, we compared for the standard FAME analysis to look at all the fatty acids and so on in the fats to compare, and they're pretty similar. There are a few differences, but actually pretty similar. And then we got IRB approval. Don't worry, we got approval to do it for the students to cook the fat and cook the real fat and our cell-grown fat and do kind of sensory analysis by smelling it and do questionnaires and all that. And the good thing is when we were all done, that's what some of the students are doing, you couldn't really, there was no statistical difference between the real thing and what we're growing is still real, but grown outside of the animal, right? That's a great starting point. And I think that's only the tip of the iceberg, because now we've got other studies we're starting to write up this summer where you can start to tailor what you put in when you grow the cells and have the cells make different compositions of fatty acids. So think of cells to grow fat that still smell great, but also are more healthy for you, as one of many examples. All right, so that's what I briefly covered. Some of these I touched on, some I didn't. I'm happy to answer questions when I get done. But the idea is there are lots of challenges still. The technology is there. The technology works. We still have work to do to get it scaled up and out to supermarkets and things like that, but it's coming. Okay, so the last few minutes I want to spend just to highlight what I would call some of the real novelties of cellular agriculture that you can't do by using traditional livestock grown meat. And therefore, I think this will be some of the driving forces for why this field will grow, and I would argue has to grow if we're going to feed the planet. So I'm going to focus more on the cell side because that's really the heart of cell ag compared to plants and so on. And so this is an older paper a couple of years ago we published. Details don't matter so much. These are pig cells. And what the PhD student did there is fed the pig cells different fatty acids. You can buy these different kinds, right? And all you have to notice here is that the bar charts are different, meaning we could tailor the fatty acid composition just by feeding the pig cells in a cell culture chamber to show that you could change the composition. That was huge. So I think it shows us we could do that. And with more recent data, you could do it with a tremendous degree of impact on what the flavors and so on are going to be for those foods. You feel free to laugh at my attempt at this slide, but the idea is also just to compare the broader aspects, right? So if you take your cow in the field or livestock, right, you're feeding whatever you feed the cow, out comes the meat and also the waste. And you're sort of bound to the metabolic profile of that animal, right? You have little control over that. And even if you want to genetically modify that animal, it's going to take you a long time to build a livestock that's able to change what they do metabolically, or even the microflora, what the microbiome is in the animal. So you're limited in terms of the degree to which you can change the meat that comes out of this animal. With cells grown in a bioreactor, everything changes. That's my point, okay? Because you have direct control of what you feed the cells, which means you have direct control of the meat. And that means what it's going to smell like, what it's going to taste like, what its nutritional composition will be, perhaps even what its health benefits might be. So think about the future of your food, tailored, personalized food to what you need from a medical standpoint, to your age, to if you're an athlete or whatever it might be, what disease you have. You can start to really think about correlating your genetics and your metabolomics to what food you're going to consume, what you're going to buy and consume that day. I think those days are coming based on the technology we're talking about today. And the other thing we already touched on, for a cow, you can make breeding changes, but I think it's 18 months to 2 years to get a cow all the way to full mature to make meat. So the process is extremely slow. Here, obviously, with genetics and so on, or just feeding the cells, it takes hours to days, and you effect a change and know if it's going to work. So I think this is a game changer. Well, time will tell. And all I'm talking about here is just feeding, right? So no GMO, but you also have the whole world of GMO at your fingertips. So here's one example we've published on, right? We've basically fused those cow cells we talked about to a carrot, okay? So you might say, Dave, why would you do that? Because the carrot is full of vitamin A, right? Vitamin A is a wonderful antioxidant. And so this is what we did. We basically incorporated the 3-step pathway that doesn't exist in the cow cell into the cow cells. And it works because you look at the cow cells, they look a little bit orange, so you can already tell that they're producing the vitamin A. And again, I didn't show all the stuff here. You can read about it if you want. But the amazing part to me is not just that it worked, because these tools are getting pretty good, but the upfront cells with the antioxidant, in principle, should be more shelf stable as a raw ingredient. So healthier when you go to buy it and cook it. And downstream, after we cooked it, we measured antioxidant levels, and they were higher because of the fact they were protected. The vitamin A is protected in the cell, so it doesn't denature as quickly upon cooking, which is really good. So lots of benefits here. The whole reason we did this was to demonstrate medically useful foods. We thought this would be good for patients that might have irritable bowel disease and other inflammatory issues in their gut, and they could just consume their food and get the benefit at the same time. That's kind of where we were trying to go with this. So these are all kind of things I see as really salient points. And then another one, if you haven't thought about it yet today, you should. And if you look at what foods you eat on a yearly basis, you're very, very limited in what your source material is, right? Chicken, cow, pork, and a few others, some fish. Why is that? Because those are the species that were domesticated thousands of years ago, right? And we're kind of stuck with those because they were easy to grow in tight, dense quarters with fairly rapid cycles. But with the tools we're talking about today, you could take a cell from any species on the planet, even endangered species. I joke because our icon at Tufts is the elephant. You could grow elephant meat if you wanted to. We haven't done it, don't worry, but you could, right? My point is you would open up not only palates, sensory, whatever, things we don't even know what they taste like today, but also perhaps cells from other species that are more therapeutically beneficial to us in ways we haven't even thought about yet. So this just opens up a whole new horizon. And I know I say these things like it's science fiction, but there's actually a company, Val Foods in Australia, that does this already. Their first product that they're scaled up already is a non-traditional domesticated food source. They decided to do something different than all the other companies. So we'll see how that goes, but that's the goal. One of the other novelties we grow in our lab is insect cells. You can skip all the details. I'll just point out that we think insect cells for cultivated meat are really, really important going forward for many reasons. Negating the fact consumers won't want to eat insects, but insects are consumed all around the world other than westernized countries, but very, very low production costs. Insect cells already don't need serum, already don't need growth factors. So they're really low cost to grow. They really don't even need any process control. We've incubated them, left them for a month, and they're all still alive. Didn't feed again, didn't do anything, right? So if you want to get really involved in something novel, this would be great. And they're also really well known to produce a lot of interesting antimicrobial peptides. So I think there's a lot we can learn here. So let me close just by saying lots of things ahead. Happy to talk more about it. I have an amazing team. Some of you here who came to the lab, you know how fortunate I am to have such amazing grad students and postdocs. So thanks to all of them. They did all the work I talked about. And I'd love to answer any questions if there are some. Thank you very much. Yeah.
Thank you so much for your talk. My name is Cameron. I'm a first-year PhD student in Kaplan lab, actually. And I was really curious when you mentioned that it could be like an opportunity in the future to replace conventional farming as like a new source of income for farmers. So I guess in your perfect world, or like your perfect vision of maybe 100 years into the future, what would that kind of look like in terms of where production centers would be located and like who would be operating them, things like that?
Yeah, great question. I see two things in the future, but maybe you can think about this better than I can because I'm not a farmer, right? I did work on a farm one summer. I think, first of all, you always need specialty animals as cultivars for sourcing cells because you want to always have ways to replenish cell stocks if you need them. But if you just followed what I said about the biomaterials, I mean, you could start to think about cropping so many possibilities and use engineered crops as well as non-engineered crops and create both media ingredients, but also scaffolding directly. And I think that can be done locally. So it could be for a local community or even an individual household to, providing sources for larger scale production. And if you do it right, I think you eliminate a lot of the transportation costs as well, which obviously add when you deal with life cycle analysis. So that alone to me is a seller, right? Because you'd have so many supply chain opportunities to feed to these production processes with the cells going forward. Okay, thank you.
What do you think about using business bioreactors to make proteins? So I worked for a company that used soy plantain and to express casein proteins. Do you have any thoughts on how that would work economically?
I don't know precisely on that, but all I know is all the alt protein companies are failing. So I know if it works, it doesn't work well enough yet. I think we need real innovation in all protein production, not using the same old host systems. I think we're stuck in the mud there, the same things we've used for 50 years, right? And so I think there's a lot of really amazing host cells that people could start to isolate and use in ways they haven't thought of before. And with the new genetic tools that are just now really at your fingertips, I think there's ways to really enhance what we already do. So I think innovation is needed is my comment. And I don't know if that particular process is cost effective or not. I can't answer that. Okay.
Personally, I'm a boneless chicken tender fan, but I know a lot of people are bone-in wing fans. Have you thought about, in terms of the texture, taste, and eating experience, how you would incorporate something like bone into this, and would you encounter the same cost and scaling issues where you need to kind of redo and come up with similar alternative methods like you have for just the meat tissue, or how would you go about creating bone in this tissue?
Yeah, so it's funny, Danny. Like I think it was 5 years ago, I had a new PhD student come to my lab and he said, "Oh, I want to work on a hybrid product," just exactly what you just said, like a chicken wing with bone and tissue. And I said no. And I only said no because we hadn't even solved individual cells yet, right? But to me, putting the bone is easy. We know how to grow bone from tissue engineering for humans. It's a no-brainer to grow bone with the right texture, you know, and and you can grow cartilage as well. So I think just translating that over to food and doing co-cultures is what has to be done. And that hasn't been done, but people are starting to talk more and more about co-cultures for many reasons. One of which is what you're talking about. So I think it's very doable. I just want to make sure we have the boneless wing in good hands first before we get too fancy. That's all. Yeah. Yes.
Really nice talk. I'm curious, when you brought up the Impossible Burgers, right? One of the consumer points is how big the label is. When you're talking about meat products like this, what does the label ultimately end up looking like? Or is there going to be a transitory period where you indicate it's a cultivated meat product, and then eventually that's the norm meat product you put on the label with chicken?
So the answer is no one knows yet. The USDA has not yet published what the labels have to be. They've been talking about it for years. We thought they would come out with the rules 2025 based on, we had held a regulatory meeting with them in September with FDA and USDA. And the inclination was it would be this year, but we haven't seen anything yet. And with the changes going on in DC, it's hard to know when. So you hear everything, right? Some states are passing laws individually to say labels have to be, there was a joke, I don't know, like 120 font, right? Because they don't want anybody to buy it, right? And some of you know, 3 states have now banned selling cultivated meat. Think about I don't know, I got to be careful of the word I use. The irony there. There's not one product sold in those states, not even a hint of a product, and yet they're already banning something that doesn't exist, which tells you about how strong some of the lobbies are related to cattle and so on. But my point is, we don't know what the labels are going to be required yet, but they're coming, and I think there'll probably be a reasonable hybrid of things. Some people say don't use meat at all. Some say you can. Some talk about how big the letters have to be. As long as there's no skull and crossbone, I think we're fine, but time will tell. I don't know what the answer is yet. So, yeah, but really important, you know.
Thank you.
Yeah.
So there's like a lot of foods out there that are very expensive, that are luxury foods, that are specialty foods. Do you think there's a market for Cell Ag to go into these specialty foods? And is it possible at those price points to offset some of the costs and the oversight caviar?
Yep. So absolutely, there's companies making caviar, and there's companies making foie gras. The main company is in France right now. So yeah, people are picking off the high-end foods to start, because that way you don't have to fight the price issue yet. So yeah, I think those are great places to start. It gets people used to technology. New technology is scary for a lot of people, right? So it gets people comfortable with it, but you know, it's still going to be expensive products. And you also get away with not having to scale huge like we're talking about here. You could scale small and still sell products. So I think these smaller targeted products like that are actually really important for the field, because they get things out there that you can point to. They get people used to it, talking about it. So say again?
The Tesla Roadster.
Yeah, yeah. And I sometimes I go wax poetically, right? Any new technology, you guys know this is here more than anywhere. Any new technology is always going to start out expensive until you scale, and then the price comes down. You can see the price coming down already, right? And the challenge here is there's been no public investment yet in this technology, right? Unlike chip making, unlike EVs, no public investment except in the Singapores, Israel, China, et cetera. So until the US decides that that's important, it's going to be slow and steady, but that's okay. It gives us time to develop the underlying research and technology to make sure it works well. So I think, we want to have the right balance. That's all going forward. Yes, please.
So before we move to the next question, we come up to the hour, so it's 5, so in theory this is considered finished. Then we can agree to be around, so you can keep.
Yeah, I'll stick around if you have other
Some of
questions. So,
the-
okay.
Yeah.
Okay.
Thank you very much.